then share my screen. Share my PowerPoint screen, sorry. Share that. All right, so if you remember from Monday, we were briefly talking about how we will sequence proteins. And the last thing we talked about was this danzyl chloride method, where you just add this chemical called danzyl chloride to your amino acids with acid, and you get this dansylated amino acid. These are fluorescent. Depending on what fluorescent signal you get, you can tell what your N-terminus is. And I said that one of the purposes of doing a test like this is that you can see how many different peptides are in your protein. We'll talk about this when we talk about proteins proper, but proteins can be made out of multiple peptides. And a peptide is simply amino acids connected by peptide bonds. Now, if we look over the whole process of how to sequence a protein, it should look very similar to how we sequence DNA, with just a few minor differences. First, peptide bonds can be connected by disulfide bonds. And which amino acid makes our disulfide bonds again? Which is the only amino acid out of the 20 we learned that is capable of making a disulfide bond. Cis. So glycine, right? Glycine has a hydrogen. Cysteine has a SH at the end. Yep. So cysteine can do that. And we need to break all disulfide bonds, because the idea is we're going to take our peptides and we're going to chop them up into small pieces and then sequence these small pieces and then put them together like a puzzle. Now, if you have a disulfide bond, what could happen when you break it into a small piece is that you'll have this blue portion. Hey, look, I have different color pens. That's connected to this purple portion. And when you sequence it, what it will look like to you is that these are part of the same peptide. But that is not the case. When you're connected by a disulfide bond, these are actually separate peptides. So we want to separate each peptide individually. So we need to break disulfide bonds. The common way you break disulfide bonds is 2-mercaptyl ethanol. You'll also see it called beta-mercaptyl ethanol, or BME. So if you see 2-ME or BME or 2-mercaptyl ethanol or beta-mercaptyl ethanol, that is a reagent that will take any disulfide bond and break it. You can make sure these disulfide bonds are broken by adding another molecule called adioacetate to make an S-carboxymethylcysteine, but that's not done all that often. Like I never used it. Usually you just use BME, and with BME around, they will never reform by themselves. If for some reason you need to take out the BME, you can add adioacetate, and that'll permanently make sure your cysteines never reform. But yeah, so make sure you know what 2-mercaptyl ethanol does. What does this reagent do? What is its purpose? So after you break your disulfide bonds, you use either chemical or enzymatic methods to take your peptides and break them down into smaller peptides to sequence them. So before we go into the question, any questions about the process, just the beginning process of how to sequence a protein? without actually talking about the methods of doing it, just the preparatory stages. All right. So to break apart the protein, you use molecules. I know the bottom is going to be cut off for you there. You use molecules called endopeptidases. An endopeptidase will, if we look at the word, endo means between, peptidase, peptide means peptide, so peptides or peptide right there. So in between peptides, and it's an ACE, so it means it's an enzyme. What this does, an endopeptidase, it will cut inside of a protein. Now, different endopeptidases cut different things, and you do not have to memorize what amino acids endopeptidases cut after. What you need to know from me is what is an endopeptidase and what does it do. But I will never ask you, like trypsin, what residue does trypsin cut after? I'm more interested, do you know what an endopeptidase is? All right. So let's take a look at some questions for our understanding here. So I have a polypeptide, and my polypeptide has these amino acids. I know it has these amino acids. I don't know what order they're in though. So I want to begin to figure out what order these amino acids are in. So let's say I use the endopeptidase cyanide or cyanogen bromide, which will cleave on the C-termini side of any methionine. So based on my amino acids up here, what is the maximum number of peptides I might be able to get if I cleave a peptide with cyanogen bromide? What is the maximum number of peptides? So I have a 4. What else do people think? We got a 2, we got a 2. What else do people think? 2. If you cut at the end, there's 2 pieces. What does CNBR do again? CNBR, when it's exposed to any protein, it will cut the C-terminal side of any methionine it finds. So any met it finds, it cuts right after that met, right at the C-terminus of the met. So this is our met, right? It will cut right here. So there's our met. I think I forgot a carbon. I think it's carbon, carbon, sulfur, carbon, but yeah, definitely it's carbon, carbon, sulfur, carbon. So if our peptide only has 1 methionine in it, then we're going to cut right after that, and the maximum number of peptides we can have are 2. So we can have 2 peptides. Okay. How about if we're using chymotrypsin? Now, chymotrypsin, let's see, it will cleave after any, so it will cleave basically after any aromatic residue. You know, your big ring structures. So based on this, if I'm using chymotrypsin that will cut after any aromatic residue, how many peptides, what is the maximum number of peptides I can expect if I used chymotrypsin? So here's a good review of your 1-letter amino acid codes. So we have a 4. Is the number next to the letter the amount of that residue? Yeah, so I'm saying I have like 1 alanine, 4 arginines, so on and so forth. 1 sec, let me close my window because there is chatter. Will cleave either on the N or C side? That is a very good question. And I believe it cuts on the C side as well. So we have a 3. We have a 6. So who can remind me, what are our aromatic residues? We got a 4. What's an aromatic? 5. Our tryptophan, tyrosine, and phenylalanine. Yes. Wait. Yeah, that's right. Sorry, I just had a senior moment there when my mind just went blank. Okay, what are the 1-letter codes for our aromatic groups? F, W, Y. Yes, so that's where I got tripped up because I forgot. So it's not T. T is threonine. Y is tyrosine. Remember, tyrosine. When you say tyrosine, say Y for tyrosine. So it's F, Y, W. And if we think about this, I have a peptide that I know I have 1 F, 2 Ys, 1 W. I don't know if they're in this order, but I know they're in there somewhere. So if I make a cut after the F, I make a cut after the Y, make a cut after the Y, make a cut after the W, the maximum number of peptides I can suspect is 1, 2, 3, 4, 5. So really, the math is the number of residues that you cut after plus 1 is the maximum number of peptides you can have. So in question A, we had 1 methionine, 1 plus 1 is 2. Here we have 4 aromatic residues, 4 plus 1 is 5. So that makes sense. Questions about that? You can have less than 5 too. If W is at the end, there's nothing to cut after, and so you would only get 4 cuts. That's why I say maximum number. All right, let me erase this. C. You are looking, oh, we do have a question. Yeah, we're cleaving after the F, Y, and W, right? And so there's 4 of those. And so if you cleave after it, so F, Y, Y, W, if you cleave after it, that'll leave you 5 pieces. Unless one of those residues are at the very end, then there's nothing to cleave after. So you're going to have either 4 or 5 pieces when you do this. So you cleave right after it. P and S aren't aromatics. P is proline, S is serine. They are not aromatic groups, even though proline does have a ring. But anyways, if you have more questions about that, please do ask, but I'm gonna move on to C. If we look at the peptide, and there are no free sulfhydryl groups, so here's another test for you. Do you remember what a sulfhydryl is? How many disulfide bonds do we think are in the peptide? Yes, 1F, 2Y, 1W. Mhm. So if I look at this protein, I find all SHs are in disulfide bonds. How many disulfide bonds do I have? I got a 2. What do other people think? Remember, what amino acid makes up a disulfide bond again? The only amino acid that can do this. One of my favorite amino acids because it has my first initial as its one letter code, C. Yep. So my name is cysteine, threonine, alanine. 2 because there's 4C. Yeah, so to make a disulfide bond, you need 2 cysteines. So cysteine number 1 and cysteine number 2 will form a disulfide bond. So every disulfide bond is made out of 2 cysteines, right? And if I know there's 4 cysteines and none of these have a free SH, which I could check with a dioacetate, if I add a dioacetate and it doesn't stick to my protein, that means all sulfurs are in disulfide bonds, which means that I have 2 disulfide bonds if I only have 4 cysteines. And D is a little bit tricky. How many different possible disulfide bonds are there? Who's good with math and can figure that out? $16? Oof, that's a lot. Nope. Yeah, D's really a math question. It's not 16. It is not 4 squared. No. Let's think about this. 4 squared, wouldn't it be 2 since each side needs a parent side? Not quite. 2 is closer than 16 though. Let's take a look. So we have four cysteines. It's not a hard math question, but it's one of those math questions. Once you understand how it works, it can be very useful in your day-to-day, especially if you're into gambling and probabilities. I'm not saying that I am, but I'm just saying it's useful in those realms. Okay, so you have 1 cysteine, 2 cysteine, 3 cysteine, 4 cysteine. Now I'm going to number them 1, 2, 3, and 4. So let's say cysteine 1 has a disulfide bond with cysteine 2. And let's say cysteine 3 has a disulfide bond with cysteine 4. This is one possible arrangement of 2 disulfide bonds. So that's possibility number 1. Okay, what if cysteine 1 is in a disulfide bond with cysteine 3? Well, that would mean cysteine 2 is in a disulfide bond with cysteine 4. That's possibility number 2. Possibility number 3. What if cysteine 1 is in a disulfide bond with cysteine 4? That would mean cysteine 2 is in a disulfide bond with cysteine 3. That's possibility 3. And that's all there is. I cannot make any new disulfide bonds without recopying a disulfide bond I already made. But if you're no good at this, congratulations for coming to the lecture or watching the lecture recording. I am not going to ask you D on the test. That's just for fun. That's because I like math. Just saying, if you were into gambling, yes. But any questions about A, B, or C, or if you want D, even though I'm not going to ask you, I'm not going to give you cysteines and say how many arrangements. Like, I'm not going to give you 10 cysteines and say how many arrangements there are, because you do need a math equation for that. Yes, each cysteine is counted uniquely. So C1 with C2, if you write it as C2 to C1, that's the same thing, right? So, yeah. And don't ask me what the equation is. I always have to look up what the equation is when I want to do that calculation. It's like n minus 1 factorial, no, yeah, n minus 1 factorial divided by n times n minus 1 factorial. I don't, yeah, whatever. We don't need to worry about it. All right, moving on. What we do need to worry about is a sequence or a method for sequencing peptides. And this is called Edmund degradation. And this was actually around, I want to say in the 70s, and it's still used because it is incredibly efficient. So let me show you what's happening. The way Edmund degradation works is that you have a compound called PITC. And in basic conditions, what will happen is that PITC will bind your N-terminus residue, and actually does right here at the C-terminus, or the C double bond O of your N-terminus. Then, if you add anhydride trifluoroacetic acid, can't talk, what will happen is that you will cut off your N-terminus, and you make this derivative. And when you add acid, you make what's called a PTH amino acid. A PTH amino acid has fluorescence. Each of the 20 amino acids have a different wavelength that they fluoresce at. And what's great about this technique is when you do it, you chop off one peptide at a time. So you can resend your peptide for another round of this. And you can sequence your whole peptide by doing this one at a time. So even if you have 0.1 micrograms or less of some random peptide that you find, you can determine what the sequence is using Edmund degradation. And what's also great, a robot can do this because it is just simply adding a chemical, adding more chemicals, getting your amino acid out and redoing it. If something is that automated, humans are smart and lazy enough to make a computer do it. So a computer machine has been made to do this. But any questions about our diagram of Edmund degradation? An endopeptidase. So what is definition of endopeptidase? An endopeptidase is an enzyme that will cut peptide bonds. That's the definition. And it will cut peptide bonds inside a peptide. An exopeptidase cuts at the very ends, and endo cuts in the middle. After you add the anhydrous trifluoric acid, the N-terminus gets cut off. Yes. Well, yeah. So you add the anhydrous trifluoric acetic acid, your N-terminus comes off, and then you just add aqueous acid to change it into what's called a PTH amino acid. I'm not going to ask you to know the structures for this. Like, I'm not going to give you a PTH amino acid and be like, what is this molecule called? But the overall process of PITC base attaches to N-terminus, anhydrous, you can even just say anhydrous acid, and N-terminus comes off acid, PTH amino acid, that gives you fluorescence. If you understand that process, and you know it's called Edmund degradation, that's all I'm looking from you. Okay, so let's see if we do understand this process. Here's a question you might see on exam 2. I have a peptide, Ser, Cis, Lu, Val, Asp. I do Edmund degradation. So my first cycle, I react with my phenolistothiocyanate, that's PITC, just in its full name, with a base. I then incubate this with anhydrous trifluoric acetoacid, and then add aqueous acid. What do I make? Normally I would have a poll. The polls were not working today. I don't know what Zoom was doing, but is it going to be A, B, C, D, or E? Trifluoroacetic acid is not an exopeptidase because it's not an enzyme. These molecules will do like self-cutting more or less. To be a peptidase, you have to be an enzyme. So you have to be a protein or RNA. Just a molecule, we would not call that an exopeptidase, but it is the same idea of an exopeptidase. It's just technically not one, right? Like, I guess you could call a paper cutter big scissors if you want, because they do the same thing, but it would be incorrect if you ask somebody where the big scissors are. So like these reagents are like an exopeptidase, but technically they're not. Wouldn't it be E? I don't know. What do classmates think? I think A2 because it's on the first cycle. E. This is why I like the poll so much, because if there's a poll, you can answer it anonymously. And I think you have more confidence if you're anonymous. So you can always send me private messages too, by the way, if you don't want other people to know, even though you shouldn't be ashamed. But I know in reality, people don't like to speak up in case they are wrong. I'm a fool and unafraid. That can be a dangerous combination, but it can also be a great combination. It depends the situation. Okay. It is E. Because remember, the first cycle cuts off the N-terminus. The rest of the peptide is intact. And you make a PTH amino acid out of the N-terminus. And so you would send this back for another round. That's Edmund degradation. Questions? All right. So the last thing, oh wait, a couple more things. Almost last thing. Can you repeat that? Sure. What happens at Edmund degradation is that the N-terminus is cut off. Here the N-terminus is serine. It becomes a PTH amino acid, so PTH serine. The rest of the peptide is left intact. It's not broken. So you can see we have our 4 amino acids left over. This will go through another round where then the cis will be cut off, then the Lu, then the Val, and then the Asp. So E is the correct answer because it works stepwise. 1 amino acid at a time, always from the N-terminus. And so what you do after you do this, so you cut your protein up to little pieces, and you then you sequence the little pieces, and you do the same thing we did with the DNA, where you look, well, probably more like a computer will look for regions of overlap. It will connect these regions of overlap together to make your peptide. So you're cutting it into a lot of different small fragments, and you're doing this with a lot of copies of the peptide, right? So you just don't do it at one copy. You do this experiment a lot. You see what pieces are overlapping. You put these pieces back together like a puzzle. Once you know your individual peptides, you can then repeat breaking your protein and not break the disulfide bonds. So these are left intact. And that will let you know where your disulfide bonds are. Because remember in our previous, in this peptide, it's easy because there's only one cysteine in each peptide. But if there's other cysteines, right, we don't know which ones are forming disulfide bonds. So we have to do an experiment where we break the peptide apart, resequence it, and see what cysteines are connected together. And then we can put our disulfide bonds in correctly. We have two strands in our example. We have a blue protein and a purple protein. And the terminology is really confusing because the blue peptide and the purple peptide together form a protein. Oh, that's both purple, sorry. One of those was supposed to be blue. They will form together to make a protein. Yeah, I drew it like DNA, but this is not DNA. That's supposed to be a protein. Do you have two of the same strands? The blue on top and bottom, but the arrow just shows one. Oh, what's happening here is that these are different experiments. In experiment 1, you broke the blue peptide like that. In experiment 2, you broke it like this. And now you're just sequencing each experiment and seeing where the overlap is to make your final peptide. That's what those arrows are showing. Different experiments where you're breaking the peptides different ways. Each experiment is using a different endopeptidase to make different cuts. That's what's going on here. That's what we're trying to show. Does that make sense? Yeah, so you do more than 2 experiments, but it's, yeah, you have multiple copies of this peptide. And each time you do this sequencing, you use a different endopeptidase to make a different series of cuts. You sequence those cuts and then try to match up pieces that overlap to make a whole protein, make the whole sequence, make the whole primary sequence. And that's what we're talking about. I forgot to mention it today. Primary sequence is the order of amino acids, which I'm going to say AA. All right. So another way to sequence is called mass spec. So mass spec is actually super cool. Those of you who want to know more, please look into Dr. Alley's analytical courses where they talk about mass spec. The idea is you have your sample and you shoot your sample through 4,000 volts and you take a protein and you more or less turn it into a gas. This gas goes through a chamber where it's hit by nitrogen and it breaks and becomes charged. These charged particles go towards more or less a big scale where based on where we hit our scale, it'll tell us how large and how charged our protein was. We get something called M divided by Z. This is mass divided by charge. So for example, if our scale measured like, you know, we have a mass of 100 and a charge of 10, those are bad numbers. 200 mass and charge of 10, then our M to Z ratio would be 20. We'd have an M to Z of 20. And what you do is you plot these peaks, or rather you get these peaks from doing this experiment, where the height of the peak tells you how often we saw this. So for example, the M to Z peak of plus 19 was saw the most. So we say we scale that to 100, and every other peak, we scale its abundance downwards. Yeah, so basically there's giant magnets here, and the charged particles will like bend based on their mass to charge ratio, and they'll strike the detector different ways. And a computer, based on how they strike, will tell you a mass to charge ratio. It's actually really super complicated. I don't know exactly how it works. I just know the basic theory we're talking about here. It won't be random. No. It knows exactly what's happening. If you want to know the exact math of what's happening, you would want to talk to Dr. Ellie. I have not looked up like how this works on like an ultimate theory level. I can just give you the overall here's how it works if you're a user, which we are. So this is what happens when you're a user of this. So and this is actually a good point. When you are using instruments in science, it is not sufficient to treat the instrument like a black box, in that it is not sufficient to put your sample in, get results out, and blindly trust the sample, or trust the results rather, because you don't know if the results are crazy or not. So you have to know basically how something works. And so this is like the level of knowledge we need to know. Protein goes in, becomes a gas, becomes a charged. When it becomes charged, it hits a detector. And based on how it hits the detector, we get a mass to charge ratio. Now, if we had time, I would teach you how we take peaks like this and we determine the size and charge of our proteins. So from this, you can determine how big a protein is just from that information using those peaks. Unfortunately, that takes me like 15 to 20 minutes to do. And this semester, we do not have time for that. So if you really want to know how this works, let me know, and I can make like a little video. Otherwise, again, for those of you who have come to the lecture or are watching this on video, congratulations. You can just cross out this problem. I'm not going to ask you question 4 or question 5. I think they're cool, but unless you're actually going to do this, which I'm guessing a small percentage of you ever would, and a computer will do this for you now anyways. It's just good to know how to do it so you don't treat machines like a black box. But any questions about, in general, how mass spec works? Because I would like us to know that, like in general, what is going on. Not that you can read these peaks and get the weight of the molecule, but just theory-wise, there we go, what's happening. The highest peak indicates how often it was seen. So basically, you count how many times that peak hits the machine, or that like, because you're sending in like thousands of identical proteins to this process, and whatever charge to mass ratio you see the most. So here we got a charge to mass ratio of 779.83 a lot. That's our M to Z ratio. If that hit the detector the most, we say that gets a score of 100. So if it hit it like 20,000 times, we saw that peak 20,000 times, we just said, this gets a score of 100. Then we rank all the other peaks based on how often they were seen compared to our top peak. So let's say this peak had a relative abundance of 80. So if this is 100 and peak 2 was 80, that would mean peak 2, it hit 16,000 times, because 80 divided by 100 is 8. I think, I'm doing math up top of my head, so it's very dangerous. So I think 16 divided by 20, no, sorry, 0.8. We'll give you the same ratio. That's basically what's happening there. The proteins can be charged already, but they're going to get really charged once they get into the gas. So they can go in charged. Then when they become gas and get hit by the N2, they just get more charged, because what's happening is that random protons are just getting knocked off, right? So you're just bombarding your protein with like gas molecules. And what happens is you're randomly more or less breaking covalent bonds for your protons. And so that's why you get charged. But yeah, it can get charged already. It just becomes more charged. And it becomes more charged, as you can see from these peaks. It will like break off in like uniform ways, where you will get just peaks that happen way more often than everything else. You can see down here we have like baby tiny peaks, but those happen so rarely, we don't need that information. When the information presented here, we have enough that we need to determine the weight and the overall charge of our proteins. The magnet picks up the gas. How does that affect the reading since gas expands and stuff versus solid when it hits just one area? Basically, you're sending gas, which is just full of proteins, and it's striking a plate, right? So we're just seeing how it strikes the plate, and how I don't think that gas expanding is going to affect that, because this is already like super hot to begin with. To make a protein into a gas takes a lot of energy. So I can't give you a good answer for that, other than it's high temperature, high temperature to begin with. So I'm sure everything has been calibrated to take care of anything that has to do with that. It's in a vacuum. It's in a partial vacuum, yeah. Again, the actual theory of how everything like works on a theory level is beyond me, and honestly beyond the scope of this class. We just need basic user information of protein goes in, becomes gas, becomes charge, hits detector, we get mass to charge ratios. That tells us the weight of our protein. That's what we need to know. I like the thoughts of how exactly this works. I think that's great. Beyond the scope for this class, beyond the scope of my knowledge, to be honest. All right, we have a couple minutes. So let's see. Yeah, I should be able to get through at least one more question. And we're talking about protein. Well, I need to change all my dates. It is not Monday, September 23rd. It is Wednesday. Yeah. So we're going to look at a little bit of evolution. And we're going to look at primary structure. Again, what is primary structure? It is simply the sequence from N to C terminus. It's what is my protein sequence when I go from N terminus to C terminus. And what's cool when you have this information is that you can compare the same proteins from tens to hundreds of species, and you can see how over time that protein has evolved. And by knowing how a protein has evolved, you can begin to determine which amino acids are important for function. So what this chart is showing you is that we're comparing homologous proteins that are related evolutionary. And we're seeing which residues are the same in different species. The exact protein we're looking at is cytochrome C. Those of you who are in the lab, this was the protein that made our solution orange in the lab, cytochrome C. So what can we learn by lining this up? So if we line up all the proteins and we see, an amino acid doesn't change. So at this position, there is always a cysteine. At this position, there's always a cysteine. At this position, there's almost always an L, there's one I. At this position, there's always a P. This will tell us this residue is critical to the function of that protein. And if you change it, that protein no longer works. We call this an invariant residue. That amino acid at that position of the protein is unique in that any other amino acid would probably cause that protein not to work and probably cause that organism to die. So that's what we can look at through evolution and seeing all of our proteins lined up. We also have conservative substitutions. So you can see here, these are color-coded. And color coding goes, yellow is hydrophobic. And this isn't uniform. This isn't something I want you to remember. I'm just telling you how this chart works. Red is negative charge. Blue is positive. Purple. No, it is blue. Blue is positive. Purple is polar. And what we're seeing here is that if the color doesn't change, but the amino acid does change, this is called conservative. What that tells us is that as long as the amino acid has the same property, it doesn't matter what amino acid is in there. So right here, where I made that mark, you can see there's red all down the line. And depending on what organism you're in, it's either E or D. What that tells us is that all that matters is that there's a negative charge there. We don't care if it's E or D, right? Same thing for blue. Let me see if I can find a blue. Well, blue looks pretty invariant, but polar, yeah, here's a good one. These two, if you look, polar more or less runs up the whole sequence, and it really doesn't matter that much what polar is there, except for that glycine. But for that S and T, you see we have some switching. So that's called a conservative, where we just care about the property of the amino acid, not the exact amino acid. Lastly is hypervariable, which means anything can go there. It doesn't matter. It's not going to affect the function. Then basically you're just looking for a spot where like all the colors are more or less there. Like this one is hypervariable kind of because it's negative, positive, polar in different organisms. So it can tolerate a lot. Here we have some negative, we have some polar. It can tolerate a lot. So we call that hypervariable. Yellow is nonpolar. So those are kind of our distinctions. One, invariant has to be that amino acid. Conservative has to be the property of the amino acid. Hypervariable can be any amino acid. So any questions about just our designations there? And what we can see through these hypervariable is just the idea of neutral drift. And I know it's cut off, but neutral drift says random mutations that don't significantly affect function. And over time, you know, we're talking about millions of years here, hundreds of millions of years. You're just going to get random mutations. Your DNA is not perfect. And we can see by looking at different amino acids, just the idea of a neutral drift, where some amino acids have been mutated, but it doesn't seem to affect the actual protein. So what that tells us is that that protein at that position just needs an amino acid. Doesn't care what it is for the most part, just needs an amino acid. Letters in the white columns. That is telling you footnotes. So there's A for that footnote. H is somewhere else. Yeah, H is saying there's no like acetyl group at the end terminus. A is just telling you the colors. Yeah. So I know it's past time, so if you need to go, that's fine. Let's see how big this question is. Okay, A and B. That should, so how many are you left? So we have some people left. So see if you can do A and B and then C, and then we'll call it a day. If I have Val, what's a conservative mutation for Val? If I have ARG, what's a conservative mutation for ARG? And site-directed mutagenesis, I can substitute, I can go from what that's saying is, let me get my pen back out. I can go from Val to Gly. That's no problem. But I cannot go this way. Well, it seems, since we are out of time, I'll just answer these quickly. So A, Val, alanine, yes. Leucine, yes. Glue, no. Isoleucine, yes. Asparagine, no. So Val, right, looks like a V. It is hydrophobic, aliphatic. So our alanine, leucine, and isoleucine. B, yes, it is lysine. Arginine is positive, lysine is positive. So that is the only one. C, that one is trickier. So valine has this for a side chain, glycine has this. So in a protein, amino acids are really packed together. Let me actually stop sharing so I'm on the big screen. Proteins are like super packed together. There's hardly any space for movement, right? So if my fist was valine and I made a glycine, which is just a hydrogen, I basically go from here and I shrink down to here, which is okay. You don't have steric clashes. But if I have a glycine and the side chain is like right here, and this is the other part of my protein, and I try to make it a valine, it would crash into part of my protein right here. You would create steric clashes. So in general, you can shrink an amino acid through a mutation. That generally does not affect the protein that much. You can't go crazy. You can't go from like an arginine to an alanine. Well, you can, but that's more risky. But if you try to increase the side chain of a amino acid into a functioning protein, that often leads to trouble because you introduce steric clashes. You now have tried to put atoms where atoms have never been before. Your protein won't fold correctly and you'll have a misfunctioning protein. So C is steric clashes. And sorry for running so long there. Just wanted to get that question out instead of pick up on that. But I'll put homework like always. I'll put this online. I will try never to go to 258 again. That is selfish of me. I should not be using that time. But otherwise, have a good day. I'll put this up. See you later. The answer to B is lysine, the positive one. Lysine. That's it. That's the only answer. All right. Take care, everybody.